So, good evening to all. Professor in Department of Plant Pathology, Dr. Karthikeyan. All the online participants who are witnessing this lecture. And apart from that, the Dean Postgraduate Studies, we also organizing along with this is the first lecture. Our Vice Chancellor is kind enough to sanction more than 16 lecture series on the endowment lecture. We started first from the eminent speakers today. So our Vice Chancellor, Madam, is keen in importing knowledge for the PG and PhD students. Those want to enrich the knowledge across the domain. The endowment lectures, which are being planned for the whole year, because the donors, many of the plant therapists and many of the donors who want to contribute for the spreading the science. We also thank those who contributed for this endowment fund. And also, I really thank all the invited speakers for the whole endowment series, because they are taken pain and come over here after the Corona time. We meet you in person. This area have a great encouragement for the young faculties and all the students to get knowledge on that. And also, we are going to cover the emerging areas, the various topics in this lecture series. The eminent lectures, because they have own domain knowledge. They are expert in their domain, and this domain knowledge can be spread across the discipline. So this will encourage the cross-disciplinary research. That is the one thing. Why to attend the pathologist lecture by other disciplines? Because now the system biology is the newer area of alignment, where you cannot understand a single system and you cannot manipulate the system, because to understand the entire system, everybody part of the system. So either pathologist or entomologist or plant breeder or biotechnology, whatever may be, they have to integrate and work. So this kind of lecture series will definitely have a knowledge base on the cross-discipline that will definitely encourage the students to take up the cross-disciplinary research and also cutting area of research. So we also have the coming lectures we have from different national institute, Indian Institute of Science or the IRI and many institutes. Apart from that, we have internationally renowned researchers, those going to deliver the lecture. So in this, I'm really happy that the first lecture, Ricardo Oliva has accepted our invitation from Tamil Nadu Agriculture University and come over here going to deliver the lecture. So with this few introduction about why we organizing this kind of lectures and also what kind of benefit we are going to get from these lectures, the TNAU will enjoy the different kind of speakers, and also it will be useful for the future reference when it will be recorded in the video format. So with this, I welcome you, sir, for this today's endowment lecture. I welcome one and all for this evening lecture, which we have a great lecture today. Thank you, one and all.
We may request our Director CPPS to give you a small introduction about today's speaker. Madam, please.
Good afternoon to everyone. So my dear friend, Dr. Sindel, Dean, School of Postgraduate Studies, and our guest speaker, Dr. Ricardo Olivia, and all the faculty members of our university, Tamil Nadu Agriculture University, student friends, both online and also offline. Good afternoon, everyone. So today we have gathered here to hear the first endowment lecture series today. So we are thankful to you, sir, for accepting our CPPS to deliver the first lecture in our university. Thank you so much for giving this opportunity. So today we are here to hear the lecture from our speaker, that is on strategies to contain bacterial diseases in modern agriculture. So here, I'm going to give a brief introduction about him. It's my pleasure to introduce our speaker, as he's a very good researcher on the bacteriology. And he has done his undergraduate studies in biological sciences at Pontificia Universidad Católica Ecuador during 2001. And also he has completed his master's in biochemistry during 2004 at Universidad Francisco de Vitoria at Spain. And he has obtained his doctoral degree in plant pathology from Swiss Federal Institute of Technology, Zurich, during 2009. And during his study period itself, he started work as research assistant at International Potato Center, Ecuador, and also in Instituto Nacional de Investigación Agraria, Spain. Later, he worked as postdoctoral fellow in the Salzburg Laboratory at UK during 2009 to 2011. He was a visiting scientist under Prometeo Fellowship to Secretariat of Higher Education, Science, Technology and Innovation at Ecuador during 2011 to 12. And he has skills and expertise in plant biotechnology, plant genetics, pathogens, disease resistance, bioinformatics, plant defense, plant pathology, fungal genetics, biotic stress, and mitochondrial DNA, with a more specialization in bacteriology. And he has worked as assistant professor in Escuela Politécnica de Ijextico, Ecuador, from 2011 to 12. Later, he worked for IRI Philippines from 2012 to 2021 in the cadre of scientists 1 to senior scientist, and he was adjunct professor at University of Philippines, Las Banos in Philippines from 2013. He was also an extension associate supervisor in University of Southern Queensland during 2020 to 21. Presently, Dr. Ricardo is working as lead pathologist under World Vegetable Center, Taiwan from 2021. Here we are having a sponsored scheme from World Vegetable Center. So under this scheme, you have conducting a demonstration trials. He has visited this university to monitor the research trials and also to conduct the field day. So we have requested him to deliver a lecture for our students. He has readily accepted to deliver this lecture on his specialization, that is the strategies to contain the bacterial diseases. This is very much essential. This knowledge will be very much useful for both the students and also the faculty members to plan our research program in global level. So I hope it will be very useful for all the faculty and students. And I request our speaker to deliver his lecture and all the student and faculty to utilize this very good opportunity. Thank you, Nandal.
Yeah, okay. I just want to make a big thanks to the university in the name of the Vice Chancellor, the Department of Graduate Studies, the CPPS, the Department of Plant Pathology, and all the departments that have been very accommodating in showing me what they're doing, lecturing me about how some of the technologies are working here in this very nice setup. I was impressed about the diversity of crops, diversity of technologies that you have been developing and the history of the university. So I'm very honored to be here today. And of course, honored to be the first lecturer of the season. I hope I have a lot of responsibility, so I will try to match. Okay, so I am happy. I also select a topic that have to do a lot of genetics and how we see the disease in terms of from the genetics perspective. And I am happy with the students that I talked today. And I learned a lot from you about how there's other alternatives or complementary approaches to genetics. So today I want to talk about two cases. First, a little bit of work on rice and what do we learn about the pathogens, and also some specific topic about tomato and what we are doing in vegetables. Just bear with me that I'm just joined the institute, so I have not much to show, but I will show whatever we have so far. So this is the most comprehensive survey of the effect of pests and diseases. worldwide in different crops. So far, we know there is around, if you see here, around 20% of loss due to all pests and diseases together. And this is not taking into account the environmental effect to the environment or human. That's something that you have a lot of a stronghold here in the university. So the effect of pest and diseases are big, and there's a lot of responsibility that we have to approach this in a sustainable manner, long term, thinking about the future of generations. Also, for us, because we are talking about pathogens that are very specialized, so they are trained to kill the plant and have co-evolved with the plant. So we think deployment also as a strategy that need to be customized. And what I'm talking about is just if we have a resistant toolkit, either by breeding or by other modern breeding approaches, we need to deploy that gene in a very customized manner, thinking about what is the environment that we are taking place, but also the pathogen that we are talking about. So these three elements need to be considered. And probably from plant pathology perspective, you are very familiar with this concept. So also this concept, you are probably very familiar, but so far what we know is that many of the pathogens that we work with in agriculture have or establish a very intimate interaction with the host plant. And they're doing this through proteins called effectors. And effector proteins are secreted in the apoplast space. They do a lot of functions, but usually they are detected by receptor proteins in a plant and plant trigger immunity, PTI. Maybe you're familiar as well with that. But also most of the pathogens, either bacteria, fungi, or mycet, even nematodes, insects, they also secrete these cytoplasmatic effectors that have so many functions. They have so many localizations in different subcellular compartments, and now we are learning every day almost what are the target areas, the functions, and how they manipulate the host to cause disease. So we need to take into account this because usually what we are doing is, having a resistant gene that is out there, and the disease or the resistant gene is overcome, and we are kind of thinking why this happened. And we need to take into account that this is a coevolution process. So we really need to think about how the pathogen evolved in that direction, and probably there. So what I was meaning is that we need to take into account where is the origin of the pathogen. That was the first key question that we want to order, and how this pathogen evolved in time, acquiring this set of virulent factors that allow him or her, this pathogen, to colonize this host, and also how the host react and develop some sort of a scheme that allow to protect themselves. So this learning the coevolution process is very key to understand how the pathogen will function in the future, and how we deploy our genes, and what is the best strategy in the genetic perspective. So today I will talk about the story on bacterial blight. Is caused by Xanthomonas oryzae oryzae, we call XOO, and is the most important bacteria of rice. Probably you are very familiar with rice in here. But the disease is vascular, so enter through the vascular system and clot the system, and the leaves are affected. Of course, less photosynthesis, so the yield is affected. And normally we control the disease using resistant genes, we call XA genes. But as you see, the presence of resistant gene is very critical, but the pathogen managed to overcome, and some populations of the pathogen emerge to overcome these resistant genes. So we are in the same problem as at the beginning. Going back to rice, this is the tree of rice from the 3,000 genomes. And what I want to highlight is that the evolution is very complex in rice. So now going to the host. And we know that most of the groups or the cultivated rice now belong to Indica or Japonica groups. And then it's important to understand also where this rice coming from in terms of domestication. What you observe there is data points of the archaeological sites that have remains of rice. And in color, you observe the timing of those sites. Okay, so basically we have two spots of domestication. First one in the Yangtze River, that probably date of 9,000 BP, is where Japonica rice was domesticated. But also we have a second spot of the culture around the Ganges River, the basin basically, that date around 4,000 BP. And probably this is where the Indica rice kind of was domesticated, or the Proto-Indica rice. Now going back to the pathogen itself, to XO, this is, we want to collect the diversity of the pathogen. And what we observe there is the collection of all the genomes that we have so far with this pathogen. It's around 430 genomes and that were collected in many of the rice growing environments across Asia. So this is also capturing the geography, but also the time of the diversity in time. So we have 50 years of collections, basically the whole green revolution up to now. So we are capturing the diversity of the pathogen to know what's the pathogen look like right now. And what we know is that there's three big lineages here in different colors. Blue, lineage 1, red, lineage 2, and green is lineage 3. And this is, when I'm talking about lineage, I'm talking about ancestral groups. But talking about modern populations, each of these lineages evolving into 12 populations and are distinguished with different colors, probably in the next graph. So now what we did is, okay, now what about if we overlap the archaeological sites of rice, where rice was grown, and the collection that we have. And we have almost 30% of our collection are within the next 20 kilometers from our archaeological sites. And you can observe here in this graph here down, that, for instance, this in the Ganges basin, many of the archaeological sites actually overlap with the collection. So this tells us two things. First, that these archaeological sites are now using as farming areas anyway. And the second one is that this opened the door for us to start to speculate about what are those populations doing there for the last 4,000, 5,000, 6,000 years. So it opens the door for us, a window in time. As I mentioned, from the genomic perspective, we did a lot of work on these 400 genomes. We try, as you observe there is 3 lineages. I'm sorry that the figure is a little bit busy, but what it represents is the, okay, yeah, sorry for that. So it represents the 3 different lineages, lineage 1, 2, and 3 populations. All the information is condensed there. Sorry for that. But what I can tell you is that we did a lot of the pangenome. We understand what are the core genes, what are the accessory genes of which population have a set of genes. And what we discover is that here in this PCA, for instance, that each population tend to have similar set of genes. That makes sense a lot. If you go through the same evolutionary process, you tend to have the same genes. And that's something that we were expecting. But what we didn't expect is this sort of analysis of selection. So if we measure the synonymous versus not synonymous substitutions in the genome and the rate per population, you start to see the specific pattern. So each population has its own pattern of selection pressure. And you can detect those signals in the genome. And of course, if you highlight some of the important genes, cell wall W enzymes, the effector gene, secretome. We have also some signaling cascade genes. Each of the population, for instance, this population 1 have a specific pattern. So we can, if we measure selection processes or natural selection, each population have its own signature. And this is telling us that each one went through a very specific evolutionary process, a specific migration pattern, a specific interact with a specific host in this kind of time of evolution. But of course, evolution is not linear. It's always recombination, kind of messing up with things and changing the course of history, like a revolution, right? And we detect this in this network kind of analysis. You have lineage 1, lineage 2, but lineage 3 have a very complex network analysis showing a very specific case of recombination. Probably what we expect or what we believe is that lineage 3 was created between lineage 1 and 2. There's some element of recombination and these lineages appear suddenly. And we can observe this in this level number of recombinant events. You can see here the population 12. Population 12 is part of lineage 3, have an unusual level of recombination events. So if we measure how many genes are shared between different populations, a pairwise comparison, you can see that population 12 have many genes belonging to other populations. So basically it's a hybrid between the lineage 1 and lineage 2, or different populations between these lineages. So the next question that we ask is, okay, now where's the origin? So where is the geographic origin? Where is the city? Where is the exact GPS data that we consider the origin? And what we did is more a wide analysis in which we select different regions. For instance, East Africa, East Asia. We have the Mekong area, South India, North India. We have the Philippines and Indonesia, the archipelagic Southeast Asia. And we did so many analyses trying to understand what are the populations, the number of populations, what are the diversity of those regions, in number of pairways, number of genes, number of SNPs, or variation itself. And what we discover is that probably this yellow area between the Ganges and the Pearl River accumulate most of the diversity. Actually, the number of populations here are the most, or the highest. The number of variations are the highest. So that means that region has something to do with the origin, because it's the diversification point. And we even narrowed down this region into using a stochastic analysis that basically measure how long the populations were kind of evolving in that region. And now we are talking about river basins, basically, not a whole region, but river basins. And what we discovered in here, you can see in this analysis, basically showing the amount of time that the populations stay there in that region. So what it's telling me is that the Pearl River are probably the Yunnan province. There's a couple of rivers there. That region, so South China, is probably the center of origin of this pathogen. And that makes sense in terms of genetic, because we have been discovering many of the resistant genes coming from Oryza rufipocon have this origin, actually are in the same region. So that makes sense that the pathogen evolved there and have contact first with some sort of Japonica population or ancestor of Japonica. In terms of time, we noticed that there is a very specific steps in the evolution or in the emergence of these lineages. This is a molecular clock analysis, and the summary of this is that lineage 1 emerged probably around 6,000 BP. And I am highlighting these numbers here. They're actually effector genes. We will touch this a little bit in the next part. But it's the first time that we observe these effector genes appearing in the population. And it's the first time that we observe a Japonica version of this effector that we call PTHX02. Again, I will talk about this later. But that means that probably Japonica was the first host in contact. So probably XO jumped to Japonica. That makes sense from a geographic perspective, from the time perspective, and also for the type of effectors that we find in this lineage. The second one happened later. Different populations have different times, but emerged around 3 BP. And this is the first time we start to notice the indica version of PTHXO2, for instance, or other alleles that are very indica related in the pathogen. And finally, is the lineage tree, remember, was a recombinant event. And look like this recombinant event just happened recently in our time, 16th century, 15th century, and so on. And there's another type of effectors there. So this is a new effector that actually is a duplication from PTHXO3 that now we call ABRXA7. But yeah, that's a. So what you observe here is a paper from Purungana in New York State, Rafael Gutaker, and he analyzed using genomics the center of origin of rice. And what he discovered is that Japonica probably evolved in China and moved west and moved south. That's the discovery using genomic data. For indica is opposite. Probably indica, according to this analysis, evolved in India, some part in India, and moved east and moved south. So that's the kind of the direction of the move. So we took the distribution of our populations, of our genomes, which are very complex. But what we notice is that for lineage 1, the center of origin, as I said, is South China, and the movement, we repeat the same analysis that Rafael Gutaker, but this time using genomes from XO instead of rice. And the movement was very clear. So this population of this lineage moved west and moved south. That's the movement. And the time coincides with Japonica movement, the distribution of Japonica, the movement in Southeast Asia, and movement into India, and probably the combination with the Proto-Indica population. But when we analyze the lineage 2 that come later, and it's coincides with the Indica move, we notice also that this move is south, but also west, east. Sorry. So that coincides with the Indica movement. And when we analyze lineage 3, the recombinant one, the movement doesn't make any sense. The only explanation here is trade. So the time coincides with the silk trade, and probably by sea or and by land. So it's a distribution that doesn't make any sense except in the trade context. So that's why for us it was very clear who is who, where did it come from, what sort of alleles they may have. I didn't touch around the genomic, the genes that we are analyzing. I will touch a little bit more. And now I will move a little bit on the mechanism. And what you're observing here is the phloem load and upload of sugars from the mesophyll cells into the SIP complex. They are doing this using the sweet transporters. So transporters outside the cell into the apoplasm, and then taking in the SIP element. The sugar travel all the way to the zinc cell and using an opposite mechanism, but this is how the plant kind of move the sugar into the system. So what we discovered actually is that XOO, because it's a vascular pathogen, and you need to understand that the vascular system is a very dry environment in the sense that there's not much sugars, there's not much minerals, there's not much of anything. So XOO need to, if you want to grow there, you need to kind of do something to extract sugars or lipids or something. What they are doing is using these TAL effectors. And these TAL effectors probably you are not aware, but I will try to explain. There are big proteins that have a central domain that is a repeat in tandem. It's 34 repeats, 34 amino acids that are repeat in tandem, and each of the repeat determine or bind to a specific nucleotide in the DNA in the promoter region of a gene. This structure that you see over here is how they fold around the DNA. So each of the repeats make a structure that recognizes a particular nucleotide. In that sense, that is a code. So the code of the DNA, the 20 nucleotides are determined as a effector binding site. So this TAL effector recognizes this set of sequence and activate that particular gene. So what's happening here is that these effectors go through the type 3 secretion system, enter into the nucleus, find a particular gene, recognize the gene, bind to the gene, activate. In this case, is called suite 13. Is a target of PTHX02, the one that I was mentioning. And the gene produces a sugar transporter, and sugar is going out. So it's leaked into the apoplast, and the bacteria can replicate. That's how XO managed to survive, grow, and move into the vascular system. So with this information, we are in an upper hand against the pathogen, because now we know how to cut the growth. So we went a little bit further into the TAL effector genes, and we did a lot of genomics. And what you observed here is all the 24 families of TAL effectors, but we are focusing only on 3. This is PTHX02, PTHX01, and PTHX03 ABR37. So we are focusing on these 3 effector genes. Why? Because they activate 3 of the sweet promoters or sweet genes in rice. Oh, okay. Yeah, this is a little bit of the distribution of the same TAL effector. It's the same, but looking at different strains. And as you see, it's a very strategy that I mentioned. So 3 genes, 5 locations, 5 CRISPR constructs at the same time. We knocked down 5 of these places. As you see, the susceptible line here, you clip inoculate the top, and the bacteria going down and grow and cause disease. However, the CRISPR lines, we clip inoculate, the bacteria start to grow a little bit, but soon after that, cannot manage to get sugars, and the bacteria die out. So it's basically what the resistance we have. And of course, we did, the next step was multi-location trials. We have a big project funded by Gates with so many partners, and we did this we are doing it in Colombia, Burkina Faso, by that time Uganda, and Philippines was also there. And as a result of this, what we noticed is that none of those lines have any trade-off in terms of yield or any characteristic that we can measure. And this is based on at least 6 experiments in different locations. So from an agronomic perspective, we believe that those lines were not affected by any of the off-targets. In fact, these lines have been sequenced, and there is no off-target effect so far. So we are very happy to kind of move forward. And in the landscape of genome editing, you know that it's changing every day. This is a 2020 publication, and from this now it's changed a lot. For instance, Nigeria and Kenya has already accepted a regulation for genome editing in which the lines that we are producing, for instance, have no regulation at all, cannot consider as GMO. And India is in the same situation. There's a lot of hope from India local perspective. So all this product that you are producing have some value from 5 years from now, and I think there's a lot of opportunities here for your research, actually. Some of the lines as well that we did in the background are very popular lines in the Philippines, in Indonesia, even in India. This is an Indonesian line that's called Sehiran Sub1, that is almost 50% of the planting area in Indonesia. So you can, and BLB or bacterial blight is really a terrible disease there. So you can see the impact that such technologies can have in big countries like Indonesia or India. In this case, we also have the accession of Vinadan 11. That is a very popular variety now in India. It's kind of one of the growers. We have that background already, CRISPR with the mutations that we run. And instead, I have to say we are working with ICAR Institute as well and so many collaborators there, and that this work will be continuing in India locally to produce some lines that can go through regulation at some point, I guess. But again, so the other question is, if we have the resistant genes in this case, how we deploy that? So what also we are interested in is try to monitor the population's change and the population, which group of the pathogens are where. So based on all the genome analysis that we did, we select the SNPs that allow us to track the pathogen in real time and very efficiently, very fast. So we have around 40 SNPs that can detect all the pathogen groups that we know so far. And if we know the phenotype of those groups, we know how they will behave against our lines or against any other resistant gene, and we can predict the outcome. And if we do this every season, then we will have the upper hand against the pathogen, which time is the most precious thing that pathogens have, because they can evolve very fast, but we cannot. So with that, we can kind of catch up with the pathogen. So we run this, for instance, to tell you about how fast we can go. We run 3,000 outbreaks in a matter of weeks from Pakistan to Indonesia. You cannot see it here, but from Pakistan to Indonesia, we have almost 3,000 outbreaks genotype in a single season. So that gives us a lot of power to deploy things that we want in the future. Also, we create a platform that we call PathoTracer. PathoTracer is meant to make a decision tool on where are the lines or where are the populations, what sort of line I need, what sort of resistant gene will be effective in this region. And it looks like this, and it's this real population data, what you observe there. For instance, if we pick here in Vietnam, this province, we know exactly what varieties or what lines can we use for breeding or for extension service, and what are the set of genes that are there. So it's a tool for breeders or a tool for extension service to kind of take decisions. So now I will switch a little bit from rice to tomato. As you know, I just joined WorldBedge, and we're trying to kind of explore what do we want to do. And this disease is called bacteria wilt. It's caused by Ralstonia solanacearum, and it's a complex disease. Many species are there, but it's also a vascular disease. And as you observe here, cause wilting. The bacteria colonize the vascular tissue and have many. It's a soil-borne disease that causes so many problems. And what we know is that it's the most prevalent disease across tomato and pepper and eggplant growing areas from Asia to Africa. So for WorldBedge, that's a priority. Yeah, I know that here in Tamil Nadu is there, but it's of course not one of the priorities. But for WorldBedge perspective, globally is like target number one. We have 5 to 6 targets. Viruses are there, others are there, of course. But as I mentioned in rice, this is the scheme that we have. Sugars are going out, the bacteria is happy, replicates and move. However, for Ralstonia is in the vascular system. And we believe that also TAL effectors are very important, but we don't know what is the TAL effector doing, what is the bacteria using, because it's not possible that the bacteria is eating anything, because there's nothing there. I mean, there's minima. So what is it eating? How it getting a nutrient? What nutrient is in its menu? So that's the big question that we have. The only thing that we know or that we expect is that TAL effectors can have some answers in that aspect. I have to say that there's a lot of research on Ralstonia. Ralstonia have other small effectors that are very good at manipulating plant processes, in particular immunity. So it's very good at, I'd say, suppressing immunity of the plant, and that's why it colonizes so many hosts. But how it does in the vascular system, that's a different story, because immunity is not very, prevalent there. The only thing is that it's dry. So you need to learn how to extract things sugar or lipids or amino acids, something to survive there. As I mentioned, there's talent. There is these TAL effectors in Ralstonia as well as XO. But it's not 24 families, it's 4 big groups. That study from the La Haya paper. But previous. So, and have the same structure. So each of the nucleotide will recognize, each of the repeat will recognize one single nucleotide in the tomato or eggplant or pepper genome and activate some genes. That's what we know. What genes, that's the only thing that we don't know. So what we did is sequence and collect all the Ralstonia genomes that are so far published and some that we sequence and try to predict what are the targets in the tomato genome. Okay, so there's ways to predict based on the sequence, ways to predict what would be the target site, the effector binding site of that particular gene. So if you have those genes, then you will be able to predict what probably the ways that the bacteria use to survive. And this is what we did. And here's just an example of the TAL effector that we have, the sequence that is of the TAL binding site here, and the gene that may be targeting. Of course, there's a lot of genes because it's not a very perfect prediction. So in general, what we obtain is 1,400 potential targets using all the database that we have of TAL effectors. But we look in a pipeline to narrow down this number. And first, what we did is select only genes on tomato that are being induced during infection. So only the genes that the bacteria really use when it infects tomato are being selected from that list. So apart from that, we have a resource of 1,500 genomes from tomato, wild material, a number of material cultivated and wild material. So we inspect in each of these genomes if there is any of those 500 genes that have mutations. Mutation like this here in dark. You cannot see, sorry, it's because, but basically a nucleotide deletion, maybe a substitution, maybe a insertion, something that tells us that the plant maybe mutate that part to kind of avoid or escape pathogen factors. So that gives us an indication of that the gene probably is important for the pathogen. If you find a mutation in a diverse collection. So to put the long story short, we find 64 of these variations. So that means 64 accessions have mutations in 64 genes. This is one example. For instance, this is a gene enzyme that it somehow the bacteria is using. We don't know why, but is activating during infection. It's involved in the metabolism between the galactose, from galactose to fructose. Why is activating or upregulating the gene? We don't know. But the only thing that we know is that we find a pimpinellifolium, Solanum pimpinellifolium, that have a 2 base pairs deletion in the effector binding site. Is that meaningful? We don't know. Maybe yes, maybe no. And like this, we find again 44, not 64, but 44 of genes that may have some level of involvement in this process. Remember that we target all the TAL effectors, so maybe there's multiple targets. There's not only one. But what is the mechanism behind? That's the one that we want to understand. What is the mechanism? And so far we have sugar metabolism as one of the candidate mechanisms. We have amino acid metabolism and lipids. All the 3 are represented in the list that we have. So we are kind of a little bit narrow from the beginning, but still we have a wide aspect. Other thing that we are doing is trying to understand the population of Ralstonia from a genomic perspective. What you observe is the result of 400 and 450 genomes from Ralstonia all over the world, including Taiwan. So you have here in in dots all the genome that we sequence from Taiwan, and we are keep sequence, so this will become bigger. But Ralstonia, what it has 4 phylotypes so far. But however, those phylotypes can also have ancestral groups, and we repeat the same analysis that we did with XO, but with Ralstonia, and we identified at least 19 different groups. There are 19 ancestral populations that can be distinguished using SNPs as well. Okay, so we want to do the same strategy of tracking down the bacteria everywhere at any time on any crop, and trying to understand what are the different groups doing. Because, for instance, if we take phylotype 1, we notice that there is 5 different populations, okay? And if you place these populations in the context of the geographies, so these populations over here, 2 of those populations are very diverse. Typical from India and Africa. So if you see this, you said, okay, the population that happened here in India with the minimal samples that we have from India is very diverse already and very different to what can happen in China, different to Taiwan, different to Southeast Asia in general, and also different to Africa. So that explains why some of the rootstocks that we use that were developed in China, Taiwan, are not working here, because the bacterial population may be different, and may be different from Africa. So we need to match the information that we have with the rootstocks resistant with the population size, similar to the sweet genes or XA genes in rice. So again, to repeat the story, sorry to be so repeated, but we are creating this platform that is called Vegemon. It looks very ugly, I would say. Later on, will looks very nice, I promise. But this is the information on Ralstonia that we have so far. Again, if we click in Benin, for instance, we will have a set of information about the phylotypes. Later on in the future, we will have all the populations about what rootstocks are working or not. And we plan this for many other diseases like virus, fungal, and try to kind of map in the geographies what are the phylotypes or the races or the genetic groups that are available there. So with that, I just, as a summary, I think I took a lot of time. So we, as a conclusion for us, XO, we understand where is the origin and how this pathogen acquire a number of effectors. Based on that information, we set up a CRISPR technology approach that help us to tackle the bacteria from the beginning, right? Without that analysis, we will never be able to generate a specific mutation that tackle those tal effectors. From tomato, again, we are making steps toward the same goal, is monitoring the pathogen population fast enough that we kind of fast track the pathogen and can deploy our genes or rootstocks or any of the resistant material or tolerant material in with more confidence. So in a way to help farmers down the road, of course, to manage the disease. And for us, also this kind of is a global strategy. So the fact that there's bioproducts that can help in the management of those diseases kind of add to other strategies as this one in the fight against these diseases. And I think that's it. We really value the efforts of monitoring pathogen, and we believe that is a very practical approach that we are taking through monitoring and taking one step against the pathogen. So with that, I just want to thank a lot of people. There's like maybe 25 different institutions that produce, help, involved in what you observe. There's collection of many pathogens in different countries. It's just a lot of coordination, a lot of talking with people, a lot of trying to convince people about the strategies. So it's a lot of work, but there's a very nice group of people behind this. And there's a lot of funding as well, Gates, USAID, PMZ, the CGR in general, MOFA, the Ministry of Foreign Affairs in Taiwan, and COA, which is the So it would be nice to have questions from the students, anything that was off. Thank you very much.
So it's nice to hear you on the pathogen evolution and the zoo isolates, how it spread across the world. My question is, for example, Rhiza sativa, we have 9,000 BP for evolution, evolved from Yangtze River Basin. And sometimes India, it is some older reports around 4,000 years of rice evolution. So when actually the pathogen and host evolution, it starts. It's a collinear at stake. Why it takes so time, more than another 4,000 BP to evolve this kind of isolates. Other it's already, it's initially while the cultivated types comes to the evolutionary first cultivation, the pathogen also came along with that, or after that it became more virulent.
Yeah. I mean, as I see right now, that's whole hypothesis from my side, right? I mean, just the pathogen evolved was a very ancestral population pathogen in South India, it's South China, the Pearl River Basin or Yunnan province. One of the ancestors of Japonica is Rufipogon. So I believe that pathogen jump into the Rufipogon population first. And when there is this hybridization with the Japonica and created the modern Japonica, domesticated Japonica, pathogen was already there. That's why we observe Japonica specific genes in the pathogen population that are very, very ancestral. Then the rice moved into India, let's say, so moved west and hybridized the Japonica population with some proto-indica that was already pseudo-domesticated in the Indian subcontinent. And then the pathogen evolved again, and the indica, modern indica appeared, and the pathogen, this new pathogen kind of came back with the indica population. So it's kind of, the pathogen moved with the rice, wherever the rice start to integrate. That makes sense, because otherwise you have to say there's two different origins of the pathogen, which I don't think is the case. Right? One in indica, one in Japonica. It's rather than came with Japonica into the indica and then in the indica to the rest of the world. And very later, I mean, in our modern time, basically, an event of recombination happened, and that produced a new population, which is called lineage 3, and actually that's the dominant one. Nowadays that's dominant. So that event of recombination kind of changed a little bit of the fitness probably of the pathogen, or make the pathogen better fit to the seed, and this pathogen travel everywhere. That's also an explanation.
Sometimes you have any idea to have some fossil records to give pathogen can isolate some DNA and work on back work on that evolutionary process. Any plan you have, or any kind of work is going on, like Neanderthal genomes they taken from fossil and come trace back some history, it's like that.
Yes, that's the very good idea. We tried to go in that direction. We didn't succeed to find specimens of rice or fossil records with the bacteria. There's a IRD in France, a group that did this, but for Xanthomonas in citrus, and because that remains in the fruit or remain in the leaf sometimes. But this is vascular, so in the vascular system is the first one to go away. So it was very difficult to recover. So we didn't recover anything, but we tried that.
Thank you.
While you are putting I want to mention something. If any one of you student is interested in Ralstonia research, we are very happy to discuss. As Dr. Shanti said, there is some collaboration between Workbench and the Institute, and there is a project behind that, and we will be happy to talk about Ralstonia or other sort of topic that you are interested. Please talk with your supervisors. If you are interested, we will be happy to discuss and to find a topic that is better suited for this location, but also from Workbench perspective, if we can help. Okay.
Good evening, sir. Sir, actually, my question is for this zoo, we have did the CRISPR-Cas technology. So we targeted the susceptible genes, sweet genes. So you have mentioned different sweet genes we have targeted, and we constructed that single guide RNA with multiplex. So my question is, when we are targeting the susceptible genes, but actually these susceptible genes will have other functions, like it will be involving in sucrose transportation and other works will be there. So whenever we are creating mutant, suppressing the susceptible genes, so what about the normal physiological activities to be function? So you have in that slide you have mentioned it was there is no phenotypical changes. So I was having that how it actually functions.
Yeah, thank you. That's a very good question actually, and that was the big fear. What we did, because this effector binding element is actually very close to the promoter region. Sometimes even touch the promoter region. You know that in plants promoter, the Tata box is very elusive term, right? You need to be careful with what you predict that is the Tata box. But usually they are very close to the Tata box, predicted Tata box. So what we did is touch the effector binding element without touching the Tata box. And one of the criteria is, that's why we want small mutations, very disruptive, that never kind of change the structure of the promoter itself. So what you want is block the pathogen without blocking the endogenous expression level. And this is one of the tests that we did to kind of make sure that the expression level of the gene is functioning normally. Because, for instance, in Sweet 11, if you touch the Tata box or you knock down that gene, the plant is very difficult to get seeds, for instance, and then that's it, right? So that was one of the criteria that we have to produce the lines, is just minimal disruption.
Thank you, sir.
So good evening, sir. It was a nice lecture in zoo rice interaction. So my question is about the executor genes. So the executor genes have been studied and they are telling it has been evolved in the rice system against zoo. So as the pathogen is also evolving, so there should be an evolution in the host side also. So what will be your idea in executor gene evolution in host side?
Yeah, I mean, executor gene is really a very nice innovation from the plant perspective, right? Basically, for those of you that doesn't know, executor gene is basically a dead domain. So if the pathogen touch or activate that gene, the cell is killed directly. So that's why we call executor. And the thing is that pathogen also evolved an innovation to get rid of the executor by deleting its own TAL effector. So the TAL effector that activates the executor is being deleted from the pathogen side. So if the executor is there, it doesn't matter. So that we have populations of the pathogen that have that particular trait, that they cannot activate any executor. So if we want to, in the future, for other crops or other pathogens, if we want to use those executor as an active dead domain, what we need to do is using CRISPR to introgress 5, 6, 7, 10 nucleotides there that are active sites of other TAL effectors, for instance, or other transcription factors that other pathogens also have, right? Just want to make clear that many pathogens activate transcription factors, sugar transporters. So it's a common mechanism. It's not XO alone, but other pathogens does do this as well. So going back to the executors, so I will take the executor itself and try to modulate the executor in a way that you create a binding site for 1, 2, 3, 4. So there's programs that you can do that, right? You take all the sequence of all the TAL effectors and come up with a common for all. So you can just play with that and introduce no more than 10 nucleotides that can be done. And that is not a transgenic plant itself in some of the regulations. And you can put on top of the promoter of an executor. So any pathogen that touch that, that's it. So that will be for me. I mean, that's kind of, yeah, that's good idea actually.
Hello, sir. This is Sendilvel from Pathology. Sir, what will be the impact of the climate change on the bacterial diseases and its mutation, evolution?
That will increase in several locations because of the access to other temperature, right? So if the temperature increase, in general, some bacteria tend to replicate faster, and if the weather is becoming more unpredictable, those bacteria tend to travel more frequent. However, and we know for a fact in XO that increasing temperature deactivate genes. From the 12 XA genes that we know, we grow in different temperatures. When we increase 3 degrees, almost only one gene survives. So that means that the genes deactivate itself. So they are not working anymore. Why? We don't know. If it is an NBSLRR, probably the folding is not complementing the function, so the protein can defold and is not active. But from the 12 genes that we test, only one survived increase in temperatures that was around 3 degrees. So I predict that the plant is becoming more susceptible. The pathogen can have more replication sites. However, some pathogens, the temperature also plays against, and they are becoming less and less virulent and less out of the cycle. So I would predict that some pathogens will emerge and other pathogens will disappear. So we need to be prepared, basically.
Thank you, Dr. Ricardo, for your nice presentation. How about to the development of antibiotic-resistant strains development? Do you have any idea on that? Because we are continuously using antibiotics against the BLB. Yeah.
Okay, let me tell you this. So, what I've seen traveling in rice fields, I've seen the use of antimicrobial antibiotics, which are banned in many countries. But if you go through the field and you see the package there, how this package came there, we don't know. So that means that these products are being used, first of all. And the second is that the rice environment usually is connected with other animal production systems. So those have connection. So the possibility of some of these or genes that are confer resistant to antibiotics is real. I don't think will affect much the bacteria itself, XO, but could happen that some of the human pathogens can acquire this and enter the rice system or other vegetable system or other crops. So that's more a worry, like transfer of resistance from other organisms into human pathogens that are being consumed in agriculture. In fact, we are right now working on a project in Dhaka that we are working on vegetable seed production system within the cities, and we want to assess the presence of resistance into the bacteria that are normally colonizing the tomatoes in particular, because there's so much worry about the fact that there is some human pathogen growing in tomatoes and vegetable production in general in the city. So we want to assess that. For XO, I don't feel that will be a big problem because there's other ways the control. But yeah, for more on human pathogens would be.
Another one thing is, what is the impact of the salinity of the soil and the bacterial diseases in the croplands. Salinity. Is there any influence positive, or what is your opinion about the salinity of the soil and the bacterial disease from the soil-borne pathogen, especially Erwinia? You see, that is now banana. It is a major issue nowadays. That problem is getting adverse, especially during the summer region. That is having some influence of the saline water. Have you come across any kind of the what is called the scenario about the salinity and bacterial pathogens in the soil?
Anything about, we haven't done any experiment on salinity. We did in flood intolerance and bacterial resistance. That we have a record and some information. But on salinity, salinity is so complex, and the plant response is so complex that I will predict that some of the mechanisms actually go against the immunity. So if the plant need to decide if it is immunity or tolerance to a particular stress, probably the plant will take out resources from the immunity system into growth and adaptation. So that won't be very good for the plant. Probably some pathogens can act on that, and that will be my prediction. But I haven't seen any report kind of specifying the mechanism about that. Sorry.
Just I have a clarification on bacterial evolution. I did some work on bacterial evolution. Usually the effector genes, which include both virulent and avirulent genes, usually flanked by some repeat sequences. Usually previously we demonstrated that these effectors, or whatever the genes, the pathogenic genes, it can able to jump from one race to another race. It is called horizontal gene transfer. So in this case, like CRISPR-Cas gene editing, we demonstrated that the race which can be able to cause resistance, it can break down to disease. So because of jumping elements, usually these gene sequences associated with flanked by insertion sequences, transposons, some page elements, so it can be able to jump from one race to another race. So it is previously demonstrated in Pseudomonas syringae, bean-citrus interaction. So in this case, like a kind of evolution, so how in future, like a CRISPR-Cas editing can be satisfactorily implemented?
Yeah, that's a good question, because this is one of the strategies of the pathogen to overcome, to adapt very fast, right? And it's proven that it's very effective to overcome immunity in the plant. Pseudomonas, even Xanthomonas, in human pathogens as well. And we know that that's why we decide to go for a starvation type or nutrient type of strategy, in which we are depriving or we are blocking the way that the pathogen feed, right? Why? Because the problem is that how the pathogen acquire a new feeding mechanism. That in evolution take a lot of time, right? Can happen that can have the whole set of a whole, new set of sugar processing genes. But that is a big evolution process. So that's why we are buying time in terms of agriculture until that happens, right? I'm not saying that this is the ultimate solution, but at least a solution that gives us a lot of time, because the pathogen will have many problems acquiring other metabolic process that allow to feed on lipids, for instance, or in other sugars that can be extracted. And that will happen using what you are mentioning, right? If they are acquiring some sort of metabolism, additional metabolism, that will happen through jumping elements, probably. That's but yeah, we have some time at least, I guess.
Thank you, Dr. Ricardo. The most respected Dean SPGS, our beloved Director CPPS, respected Controller of Examinations, and our today's speaker, Dr. Ricardo, heads of the departments, my dear colleagues, student friends, research fellows, good evening to one and all. On behalf of SPGS as well as Directorate of CPPS, first of all, we would like to thank our Honorable Vice Chancellor for permitting us to conduct this endowment lecture. That too, we are of in a great thing that the first endowment is organized in the series, endowment series. So that also we need to thank our Dean SPGS for giving us the opportunity. Thank you, sir. Thank you very much. And also we thank our Dean SPGS and also Director CPPS for their continuous motivation as well as the arrangements for arranging this endowment lecture. Thank you, madam. Thank you, Dean SPGS, sir. And also, profusely, we need to thank our special guest lecturer today, Dr. Ricardo. Though he has come here to review the progress of work of the IPM project under the sponsored by World Wage Project Taiwan, and he is able to give the lecture and give the timing to give this endowment lecture. And this lecture was highly useful. You talked about two different major bacterial diseases. Though we are strong in other pathogens like fungi as well as the viruses, even phytoplasma, but as far as the bacteria work is concerned regarding the management, it is very, very less in our university. So with your informations and the strategies, we would like to continue and start to work on the bacterial diseases. So the different approaches, especially the molecular strategies you proposed through gene editing, like CRISPR-Cas or whatever it may be, it will be a highly targeting the effector genes. It will be a highly useful. So we will take up the research. Thank you very much for your interesting and thought-provoking talk on the host pathogen interaction also, especially the phytopathogenic bacteria with these two hosts, especially rice and the tomato. Thank you very much for your nice speech. And I thank Controller of Examination for his presence here in this evening. Thank you, sir. Thank you very much. And I also thank the President of Agriculture Entomology as well as the Nematology and the fellow friends of the Department of Plant Pathology and other departments, and also the student friends and JRF, SRF, and other research fellows, postdocs who are attending both offline as well as online. And I profusely thank the help rendered by Dr. M. Murugan, Professor Agriculture Entomology, Dr. K. Angapan, Professor Plant Pathology, as well as the PG coordinators of Department of Plant Pathology, Dr. V.K. Parthiban, as well as the Dr. I. Johnson. And also the help rendered by the professors and other staff of the office of this Dean SPGS is also acknowledged. Thank you very much. Thanks again. Thank you.